Hello everyone, welcome to my channel Botany for Neet. Let us solve a question asked in Neet examination. In angiosperm, flowering plants basically, the haploid, diploid, and triploid structures of a fertilized embryo sac sequentially are synergids, primary endosperm nucleus and zygote, antipodals, synergids and primary endosperm nucleus, synergids, zygote and primary endosperm nucleus, synergids, antipodals and polar nuclei. So basically, they're asking the ploidy whether which of these cells are haploid, diploid, and triploid. Before solving the question, so here it's a diagram of pistil or carpel, which is the female reproductive organ in a flower, and it comprises of ovary and this tube-like structure called style, and the receptive part known as the stigma. When a compatible pollen grain falls onto the stigma, the pollen tube is formed. This pollen tube elongates and reaches the egg cell present inside the embryo sac. This is the embryo sac which is present. Embryo sac is also called female gametophyte. It develops inside the ovary. Okay, let us look into the structure of this female gametophyte in detail in the next slide. But actually, the female reproductive organ is carpel or pistil, and it has three parts: ovary, style, and stigma. If we consider a hibiscus flower, if the pollen grains of the hibiscus flower fall on the stigma of the hibiscus flower, then there will be compatible pollination. If hibiscus pollen grains fall on rose stigma, then obviously it's not a compatible pollination. So that's when I mean compatible, that means the pollen grains of one plant kind is falling onto the stigma of the same plant kind, resulting in the elongation of the pollen tube and meeting it at the female gametophyte, also known as embryo sac. Now, if you see, this is the embryo sac, also known as female gametophyte. Okay, where now you can see here there is like cell, two synergids, polar nuclei, and two antipodals. Reverse is shown in this diagram. Down you have antipodal cells. There are three cells, antipodal cells, one central nuclei, one egg cell, and two synergids. Total how many cells? 2 plus 1, 3, 3 plus 1, 4, 4 plus 3, 7 cells. But here, if you see, these three cells have three nuclei. Okay, but this one cell has two nuclei, and this one egg cell has one nuclei. These two synergids have two nuclei. Now, can you tell me the total number of nuclei? 2 plus 1, 3, 3 plus 2, 5, 5 plus 3, 8. 8 nuclei. That's the reason female gametophyte, in case of angiosperms, it is described as seven celled, eight nucleated embryo sac. In this way, it is usually described seven celled, eight nucleated. I hope you understood why it is called seven cell. Three cells, 3 plus 1, 4, 4 plus 1, 5, 5 plus 2, 7. Okay, seven celled. Here it's explaining how it is seven cell. Okay, I'm sorry. So, two. Okay, so two cells, one egg cell, here one polar nuclei, three cells, total seven cells and eight nuclei. Now, after fertilization, what happens is basically the pollen tube, it enters and releases its male gamete. This male gamete fuses with the egg cell, and then fusion of the male gamete with the egg cell results in formation of a diploid cell known as zygote. Because egg cell is haploid because it is a product of meiosis. Male gamete is also haploid. It is also a product of meiosis, which is formed from microspore mother cell and megaspore mother cell. So zygote is diploid. Okay, so zygote which is formed, it is diploid. Now we know that there are two male gametes which are produced, right? So one of the male gamete fuses with egg, while the other male gamete it fuses with the central cell, which is having two polar nuclei. So what's happening? One male gamete fused with egg cell, while the other male gamete fuses with the central cell, which is having two nuclei. Okay, so this results in formation of a triploid structure known as primary endosperm nucleus or endosperm. So fusion of the male gamete with the central cell results in a triploid structure pen where it serves as a nutrition for the developing embryo. Okay. So, in angiosperms, that is flowering plants, haploid, diploid, and triploid structures of fertilized embryo sac. After fertilization takes place, we know one of the male gamete fuses with the egg cell to form zygote, which is diploid structure. Zygote is diploid. Other male gamete fuses with the central cell to form primary endosperm nucleus, which is triploid. Okay, and we know these structures, basically your antipodals and the synergies which are present on either side of the egg cell, they are haploid. Prime polar egg cell is haploid. Okay, pollen grains having the male gamete is, it is also haploid. But when the egg cell fuses with the male gamete, it results in formation of zygote, which is a diploid product. In the same way, when the male gamete fuses with the polar nuclei containing two nucleus in it, the polar central cell containing two nucleus, then it results in formation of a triploid pen, primary endosperm nucleus, which serves as nutrition for developing embryo. While the synergids which are present on either side of the egg cell or the antipodals, they are haploid. So the best option that describes the answer is option 3, where haploid structure are synergids, diploid are zygote, and triploid structure in the embryo sac, fertilized embryo sac, is primary endosperm nucleus. I hope the solution for this question is clear. If you have any doubts, please feel free to comment down below. Thank you. Have a good day. Happy learning.